I will be talking about the second part of the lecture, which is on chromosome analysis and structural aberrations. Now, in the first part of the lecture, we discussed about the karyotypes. Now, if we see, a karyotype is basically an individual's complete set of chromosomes which is mapped. So, the karyotype is basically a name which refers to the laboratory produced image of a person's chromosomes which are isolated from individual cells, and then they are arranged numerically, which means that the first, second, third, fourth, like this chromosomes are arranged. Now, these karyotypes helps us to understand or look into the abnormalities of the chromosomal number or the structures which are there. Now, the karyotype plays a very important role in the cytogenetic testing and the checking up the change in the chromosomal number. Now, there is a term which is there which is called as the aneuploidy. Now, the aneuploidy is basically a term which is used when there is increase of one or more chromosomes or there is a missing chromosome which is there, which means that apart from the normal 46 chromosome, the person may have 47 chromosomes or 48 chromosomes, or there may be a missing chromosome, which means that the person may be having 45 chromosomes also. So, the karyotyping helps to do the cytogenetic testing, and it helps to determine what are the chromosomal abnormalities which are present or the structural abnormalities within these chromosomes which are present. Now, there are various aneuploidy conditions which are there which have been recognized. For example, the Down syndrome where there is trisomy 21, Edwards syndrome which is trisomy 18, Turner syndrome monosomy X which is there. So, now these particular aneuploidy, there are, apart from that, there are structural abnormalities also which are there, which means that the structure of the chromosome is not proper or intact. So, there are various forms or the ways due to which it has occurred, which includes the duplication, deletion, translocation, inversion and insertion. We will come into each of them one by one with the help of the images over there. Now, how exactly these karyotypings are helping us nowadays? Now, these karyotyping plays a very important role now in the diagnostic tools for checking the fetal and the genetic conditions. Now, they play a very important role nowadays for the oncology and the hematology exercises also. So, they are helping the doctors to diagnose and classify the leukemias, help them to estimate the prognosis, the treatment regimes and the response to the treatment with by taking those particular drugs or the medicines over there. Now, how exactly a karyotype is being prepared, which is shown over here. So, the blood sample of the person or the patient is taken up, then subsequently it is being treated and then it is cultured over a period of time. After culturing, what happens is there is an addition of the colchicine which is added to these particular culture plates, so that all these dividing cells, they stop at a single phase which is called as the metaphase. The metaphase is being used because the chromosomes are best studied in the metaphase condition because of the condensation and the compact structures they have. Now, once the metaphase arrest has occurred, then subsequently the various chemical treatments like the hypotonic solution treatments are given up so that the cells bulge out, and once they are dropped onto the slide, they burst out. So, all the chromosomes of that particular cells are dispersed in those particular slides. They are then stained with the help of the various stains. Like for example, one of the basic stain is the Giemsa staining, which gives each of the chromosome characteristic banding patterns, and then the images are taken up and then they are arranged according to the chromosome number. Now, the chromosome banding techniques helps to produce a series of consistent landmarks along the length of the chromosomes. So, with the help of these particular landmark chromosomes mapping, we can easily identify whether the specific segment of an individual have been deleted or translocated, or there is an inversion which has taken place or not, along with the help of the standard plate images which we have. Now, these are the various banding patterns which are shown over here. So, we can see the various staining techniques which are shown over here, the Giemsa staining, and then the Q binding staining, the R binding and the C binding staining over there. So we can see over here each of the chromosome pairs are being arranged in a sequential way, and then they are observed whether there is any kind of chromosomal aberrations or the structural changes which has occurred or not. Now, the chromosomal abnormalities can be numerical or it can be structural, which means that there may be increase or decrease in the chromosome number, or there may be changes in the structure of the chromosome. The structure of the chromosome can change with the help of the various methods, like for example deletion, duplication, inversions, translocations, like this one. While in the case of the numerical abnormality, what happens is one of the chromosome of a pair can be missing, or one or more chromosome number can increase also in these particular thing. A structural abnormality, I said that the chromosome structures have been altered in one of the several ways which are present over here. Now, why exactly these chromosomal abnormalities occur? Now, there is a phenomena which is called as the non-disjunction of the chromosomes. We know the methods of division, which includes the mitosis and the meiosis condition. Now, in the mitosis condition, we know that the two cells that are duplicates of the original cell. So, one cell with the 46 number chromosome, it divides to become two cells exactly duplicate of that one, which is 46 chromosomes each in each of the cells. So, this is called as the mitosis. While in the case of the meiosis, it results in the daughter cells which are half the number of the chromosomes, which means that there are 23 chromosomes are present instead of the normal 46. Now, this kind of division occurs in the reproductive organs, resulting in the eggs and the sperm. So, the eggs and the sperm chromosomes basically contains half the number of the chromosomes, because when they fertilize with one another, they form a normal cell which will be having 46 number of chromosomes. So, that is why in the meiosis, it reduces to the half the number of the chromosomes over there. Now, the structural abnormalities of the chromosome can be occurring with the help of the various processes, like for example deletion, duplications, translocation. Now, deletion means a portion of the chromosome is missing or deleted over there. Duplication, a portion of the chromosome is duplicated, resulting in an extra genetic material, while translocation, a portion of one chromosome is transferred from one chromosome to the other chromosome over there. Now, in the case of the inversion, a portion of the chromosome has broken off and turned upside down and reattached. So, as a matter of the fact, what happens is the genetic material has inverted. That is why it is called as the inversion. Now, we see the diagram over here. So, these are the diagram which shows the examples of the chromosomal abnormalities which occurs over here. Now, if we see over here, the deletion has occurred, which means that a part of the chromosome has been deleted. So, we can see over here the chromosome length has decreased or shortened in these particular cases. Then comes the duplication, where a part or the segment of the chromosome duplicates, or the copy number increases. So, what happens is the chromosome size increases because there is a duplicate copy of that particular gene which is present, and finally the inversion, which means that a part of the segment of the chromosome, it has inverted, means it has flipped upside down. So, there is an inversion, and then comes the next one, which is called as the translocation. So, a part of the segment of the chromosome, from one chromosome it moves to the other chromosome. So, this is called as the translocation over there. So, these are the various examples of chromosomal abnormalities which are occurring. Now, over here we will touch upon an example of the deletion which has occurred. So, there is a syndrome which is called as the Cry-Du-Chat syndrome. Now, the Cry-Du-Chat syndrome is a rare genetic disorder where the variable portion of a short arm of the chromosomal 5 is missing or it is deleted. Now, what happens is, due to this particular deletion, there are various changes which occurs in that particular patient or that particular child. Now, why the name has come up? Because the child has got a distinctive cry which resembles the meowing of a cat. Now, there are various facial feature changes which occurs in those particular due to the deletion. The growth is slow. Then there is microcephaly which occurs, which means that the head circumference is smaller than what you expect in a normal infant age and sex in those particular cases. So, we can see over here how a small deletion is occurring, which is causing a changes in that particular patient on the child over there. Now, there are two main types of translocations which are there. One is called as the reciprocal translocation, the other one is called as the Robertsonian translocation. Now, in the reciprocal translocation, the segments from the two different chromosomes have been exchanged. So, we can see over here in the image, the segments have been exchanged. While in the case of the Robertsonian translocation, an entire chromosome has attached to another chromosome at the centromere. So, this is a change in the chromosomal aberration or the number which occurs over there. Now, why exactly the karyotype analysis is important? Now, in the karyotype analysis, first it involves the counting of the number of the chromosomes, which means whether the person sample has got the appropriate number of chromosomes present or not, and once it is being done, then the careful analysis or examination of the binding pattern of each of the individual chromosomes in the selected cell. So, this is what is being done in the karyotype analysis or the chromosomal analysis. Now, the binding pattern of each of the chromosomes is very specific and it shows a characteristic feature which helps the scientist to arrange them in a karyotype or a ideogram. Now, the cytogenesis analysis, they analyze each pair of the homologous chromosome which are directly looking under the microscope or taking an image of that particular thing and then comparing with the standard forms. Then they can decipher out what are the chromosomal changes which has occurred. Now, this is an ideogram which is shown over here. So, an ideogram showing the binding patterns of the individual chromosome as revealed by the fluorescent or the Giemsa staining over there. Now, there are various binding patterns which are there, which one of them includes the G binding pattern. So, if we can see the image of the cell over here, the chromosomes are scattered over there, and if we see each of these chromosomes, it has been stained with the help of the Giemsa stain, and we can see the characteristic spread of these cells, and the images are being taken up, and subsequently what happens is each of these chromosomes are then arranged numerically to check the presence of each of the chromosomes, and then they further analyze each of the chromosome binding patterns, whether there is some kind of chromosomal aberrations or the structural changes have occurred or not. So, this is how a metaphase spread looks like. Now, one of the advanced technique which has come up for the chromosome analysis is called as the chromosomal microarrays also. Now, many a times the chromosomal microarrays are referred as the cytogenetic microarray, molecular karyotyping, or the genomic copy number arrays which is there. So, this is basically a DNA based genome wide testing method to detect and categorize whether the patient sample is having any kind of DNA duplication or deletion which occur or not. Now, this is a very advanced level of information which is there. So, it has revolutionized the medical genetic diagnostic practices across the globe. Now, if we see the diagrammatic representation over here, this shows that each of these microarray plates, they contains the genome or the all sets of chromosomes which are present. Then the probes are being inculcated over there, and they go and bind to those particular areas over there to check whether there is some kind of deletions or duplications which are there, and then these microarray plates are inserted into the specialized machines which analyze the images or the fluorescent label probes images over there, and they give the exact location in which chromosomes and which part of the chromosome the aberration, structural or the numerical things have occurred over there or not. Now, another level of the advanced technique is called as the fluorescent in situ hybridization, which is many a times referred as the FISH. Now, the fluorescent in situ hybridization is basically a molecular cytogenetic technique which helps to localize or find out a specific DNA sequence within that particular chromosomes in a cell. Now, for this particular analysis, it uses a probe which is basically a purified form of a DNA which is tagged with a fluorescent dye. Now, this probe will go and bind to that particular complementary part of the chromosome where it has to get attached over there. Now, the fluorescent label probes, it finds and then binds to the matching sequence within the set of the chromosome. So, with the help of these probes and specialized microscope, the chromosome and the subchromosomal location where the fluorescent probe has bound can be easily checked and analyzed. Now, these particular things help the scientists to go for a molecular cytogenetics of the cells over here. Now, if we see the image over here, this image shows how exactly the FISH is being done. So, the double-stranded DNA, it has been released. So, the single-stranded DNAs are formed, then the probes are added over there which goes and binds to the specific location within these particular DNA, and then these probe analysis is being done because the fluorescent label images are being released which can be easily checked under the microscopes. Now, the in situ hybridization technique was initially developed in the year 1960s by a number of scientists' collaboration, and it has become a very powerful tool for determining the chromosomal location of the hybridized nucleic acids over there. Now, soon after the fluorescent labeled are being attached over here, the fluorescent labeled over a period of time, what they have done is they have replaced the radioactive labels because the radioactive labels didn't have had safety concerns which were there. Secondly, about the stability and the ease of detection. So, on the basis of all these parameters, the radioactive labeled have been replaced with the help of the fluorescent labeled nowadays. Now, if we see the image over here, this is a FISH which is being shown, which is showing the identification of the human chromosomes through the chromosome painting. So, we can see each of the chromosome patterns is a specific patterns and they are glowing with the particular images because the fluorescent probe has been tagged with a particular dye over there which is emitting the fluorescence and which can be taken up with the help of the specialized instruments or the microscopes, and this is how we can decipher out and find out the various chromosomes within a person. Now, there are various chromosomal abnormalities which are there, which can be the numerical abnormalities or the structural abnormalities. So, we will come into some of these abnormalities over here and we will discuss about how exactly these chromosomal abnormalities are resulting in the disease conditions. Now, before going into that one, let us take up why exactly these abnormalities are occurring, because it takes place with the help of a process which is called as the non-disjunction of the chromosome. Now, this process is basically the failure of the chromosomes or the chromatid arms to get separated into the daughter cells during meiosis or the mitosis process. So, with the help of the images, we will see how exactly the non-disjunction are occurring. So, the non-disjunction in the case of the mitotic generally occur in the early cleavage cells. So, there will be a mosaic cells. So, one will be having a larger number of chromosome while the other will be having a lesser number of chromosome because of the non-disjunction of the chromosome. So, we can see over here an image in which we can see, if we see the first image, so we can see instead of each of the chromosomes going into either of the daughter cells, in one of the cases what is happening is both the chromosomes are going into one daughter cells, while the other daughter cell is not having any of the chromosomes. So, this is called as the non-disjunction of the chromosome. Now, if we see in a better form over here, we can see the non-disjunction which is occurring over here, which can occur in the chromosomes also or in the chromatid arms also, the non-disjunction. Due to this thing, what is happening is there is a mosaic which is formed, which means that there is an uneven distribution of the chromosome or the chromatid arms within these daughter cells, and resulting in the disease conditions over there. Now we will come into the first disease condition, which is called as the Patayo syndrome over there. Now the Patayo syndrome is a serious rare genetic disorder which is occurring. In this one, what happens is there is an additional copy of chromosome 13. So many a times it is referred as the trisomy 13. Now the baby with the Patayo syndrome has got 3 copies of chromosome 13 instead of 2 copies. So due to the non-disjunction of the chromosomes, what is happening is instead of 2 sets or 2 copies of a chromosome 13, the baby is having 3 copies of chromosome 13 within the cells. Now this particular condition, it severely disrupts the normal developmental process in the baby, and it has been seen that it affects about 1 in every 5,000 births, and 9 out of 10 children born with Patayo syndrome die during the first year of the birth actually. Now in the trisomy 13, which is many times referred as the Patayo because it was first clinically identified in the 1960s by the scientist Dr. Patayo, he published a paper. Now in this one, what happens is there are 3 copies of chromosome 13 which is there, which is mostly because of the non-disjunction in the meiosis process occurring frequently in the mothers of advanced age greater than the age of 35 and 40 actually. Now the clinical syndrome of the Patayo syndrome were initially characterized as the cerebral defects which were there, cleft plate which was there, hair lip which was there, polydactyl which was there. Now the long surviving patient with the Patayo syndrome has got number of cerebral and the cardiovascular malfunctions which are also there. Now if we see the image over here, the baby is having a lip cleft which is there. So there is a cleft which is formed over there in the roof of the mouth, which is many times referred as the cleft plate over there. Now the baby has got heart defects also, there are brain and spinal cord abnormalities which are there, poorly developed eyes which are there, extra finger or toes which are there, kidney defects are there. So these are the common medical problems which are included in the Patayo syndrome in the babies. Now if we see the karyotype image of these babies, so we can see one of the chromosome numbers has increased over there. So the chromosome number 13 is in trisomy. So 3 chromosomes of chromosome 13 are present over there, due to which these kind of conditions are occurring. The next one comes the Edwards syndrome. Now the Edwards syndrome is basically the trisomy 18, which is a rare but serious condition. Now most of the babies with the Edwards syndrome generally die before or shortly after being born actually. Now the Edwards syndrome, basically babies has got 3 copies of chromosome 18 instead of 2. So again there is a numerical change in the copy number of the chromosomes which are occurring, which is mainly due to the non-disjunction of the chromosomes which are there. Now the various syndromes of the Edwards syndrome were initially deciphered or reported by the Dr. Edwards in the 1960s, where a multiple congenital malformations and the congenital defects which were identified. So we can see there are number of congenital malfunction and congenital defect, cognitive defects which are present in the Edwards syndrome suffering baby over there. Now some of them have been referred over there, and Edwards syndrome, as again I said that it is a trisomy 18. So there is a decreased muscle tone which is there, low set of ears, internal organs are forming or functionally different over there. There is an intellectual disability which is there, which may be very severe in many of the cases also. Small physical size like the head, mouth, jaw which is there, and minimal response to the sound. So there are number of characteristic and the medical features which are associated with the Edwards syndrome over there. Now Apart from that, there are congenital heart diseases, kidney diseases which are there, breathing abnormalities which occurs, gastrointestinal tract or the abdominal wall issues or the birth defects which also occurs over there. The next comes the Down syndrome, which is the trisomy 21. Now, the Down syndrome was initially described or reported by an English physician. His name was John Down in the year 1866, and it is basically related or associated with the chromosome 21. Now, what happens is, again there is a trisomy of chromosome 21 occurs in the Down syndrome. So, there are number of chromosomal abnormalities which are occurring in these particular patients. Now, it is mostly frequently occurring in the live-born aneuploidy, which is mainly because of the trisomy 21, which causes various syndrome. Now, these occurs about 1 in 319 to 1 in 1000 live birth cases in the different population. Now, if we see the images over here, this is a baby who is suffering from the Down syndrome over there. So, we can see many of the characteristic features which is associated with the Down syndrome or the common traits which are associated with the Down syndrome. So, it includes the Down syndrome includes the different abilities. Usually the IQ level is mid to moderately low in the children. They are slow to speaking than the other children also. And some of the characteristic feature are the flattened faces which are there, and the eyes are slanted, short neck which is there, small ears which are there, small hands and feet which are there. So, these are some of the physical features of the Down syndrome suffering children over there. So, in this part of the lecture, we discussed about the various abnormalities, chromosomal abnormalities which are there, which are related to the chromosomal change in the numbers as well as the structures over there. So, and how exactly the karyotyping and the chromosome and genetic information or the analysis is important in finding out or looking out for these particular abnormalities in the children or the adults, and these are being now used, these karyotyping and the cytogenetics tools are being used to decipher out and find out or prognosis of the various diseases, for example the leukemia and the various hematology related diseases. So, with this note I come to the end of my lecture. Thank you very much.